inquire whether our co-chair, Dr. Pooja Khare, are you there, madam?
Good afternoon, sir. I'm there, sir.
Okay, good
Thank
afternoon. Welcome. And then conveners, Dr. Kaveri Mahanta and Dr. Ratna Sahai. They both must be there, and kindly help me in faithful recording of the proceedings of the session. So please make the minutes of the session. And I will not lose further time, and I will invite our co-chair, Dr. Pooja Khare, if she wants to say something on this particular session. Dr. Khare, please.
Thank you, sir. I'm thankful to the organizing committee of ANRCM and also Dr. Arora for inviting me for this occasion. And I'm not wasting much time on my talk. So let's start the session, sir.
Okay, thank you. Thank you, madam. We will interact in the meantime during the discussion and other things. So let me welcome Dr. Professor Ramchandra, and sir, I invite you to kindly deliver your talk. Please restrict to 20 minutes time limit. Professor Ramchandra.
Good afternoon, sir, and I am very happy to be in front of you all, and I am thankful to Professor Sanjay Arora also for organizing this meaningful conference and inviting me as a lead speaker. I think I am audible. I am audible. I am audible.
Yes, sir. Yes, sir.
Okay, thank you. So, good afternoon to chairperson and co-chair and other convener of this program. Please remove the disabled participant screen sharing so that I can share my slides.
Okay, sir. Now you can.
Please. First slide. Yeah. As my topic is concerned, that pulp paper industry in India and abroad world over is one important industry which consumes lot of the natural resources and discharge lot of the pollutants which matters for the soil as well as agricultural crops both. So on this occasion of the conservation and sustainable use of the water resources, this is a very important aspect under the bioresource conservation and management topic. As if you will look the pulp paper industry as whole, this is one very important industry which consumes more than 200 cubic meter of the fresh water, and accordingly it discharge more almost 200 cubic meter of the effluent after all the treatment or the product formation. In this slide, you can have a view of industry which are discharging a continuous huge amount of the effluent, which is containing lot of the chemical as a pollutants. And similarly, in the side picture, another industry is showing the discharge. If you look the view of the paper industry, this is just a view for the kraft paper, this is for the white paper, and this is a view how this discharge in form of canal is coming. This is a from different place of our country itself, where I have visited. If you will look the global scenario of the pulp paper industry, approximately 400 million metric ton of paper is produced and consumed globally each year, and among which India produces almost share 8.3% with consumption of 12.7 million metric ton of the paper. And if you look the domestic as well as the import component of the this paper, this actually is produced in India almost 11.4, while rest of portion is imported from outside. So the Indian paper industry account about global 3.7% of world production of the paper. So this is the actually seen, which is the system globally regarding the pulp paper industry, and it turnover almost in Indian currency is 60,000 crores, and almost it provides a good amount of the employment of the people, and it directly more than 0.5 million people directly and 1.5 million people indirectly as employment. So the this is the statistical data of the different country. China is on the top, USA is second position, and India comes at 15th position. So globally, India stands at 15th number in production of the paper. If you look their location in country, almost small and large scale industry calculating around 859, where larger industry has more than 25. And if you look the view of natural resources and pollution parameters in country, you can have a view how it is causing health hazards as well as the depletion of the natural resources and contamination to the environmental system. Uh, though there is the treatment facility working in the industry itself, but that is not up to mark. Therefore, they are still discharging the contaminated water. So this is directly causing health hazards to human and aquatic resources and our cattle, how they are affecting to the food chains. Yes, this is the view how the agricultural crops and soil is directly being affected nearby the industry due to mismanagement of this wastewater. This is from the western UP side near Saharanpur area, Star Paper Mill. How you can imagine nearby the industry, this is the causing pollution to the surrounding area and causing the dangerous position to the soil, and this contaminated water is also going to the agricultural crops. Not only that is affecting to the surface water as well as the soil, it is also affecting to the underground water, and this yellow containing water is having nearby the industry, and this 100 feet or 40 feet below the surface, the water is also containing lot of the pollutants, and it needs a proper investigation. And this also cause this is not a mountain, this is as a byproduct from the industry, lime. Due to the not proper recovery of the lime, it has been dumped here, and this is the view nearby industry how the water contaminated is coming from the below underground water. So what is the deficiency or gap between the policy makers and industry and the public? So the major gap is this: in the chemical property of discharge pollutants are not completely known. Their environmental effect and health hazards is not known. Adequate treatment facilities for detoxification is still not available in industry, and the status for permissible limit of industrial wastewater for various pollutants parameter are not meeting out, and role of regulatory agency is doubtful. Impact of the groundwater quality is not properly recorded so far. Coordination, there is lack of coordination between industry researchers and regulatory agency and policy makers. So this is the gap where the India and research people, academicians, and from different sectors, policymakers has to think over. If you look the physicochemical property of the wastewater, water generated from kraft paper and pulp paper industry, you will see the BOD, COD parameter and other total suspended solid are quite high, while the required limit for this permissible level is quite below. And along with this physicochemical property, there is a lot of significant amount of the metals also. So this actually enhance the toxicity of the discharge wastewater, and it also creates a bad health to the soil. This is the same things has been collected from different other places of the industry. During the analysis part, I found that most of the compounds or pollutants present in the wastewater discharge is having under this in between the UV range are in between to 3 to 400 uh, nanometer. So means they are under this category various pollutants. We try to analyze these elements and other property of the pollutants present in the discharge waste. We found the interesting point that there is a lot of the organic pollutants and playing as role of the endocrine disrupting chemicals in the nature, and they are directly or indirectly they are affecting to the natural resources like the fish or small other animals or some other small plants or animal, they are playing role in the maintaining the ecosystem. So that is causing health hazards directly to them, and that is not causing adverse effect. They are also affecting to their genetic level. So I try to investigate them through the GC mass analysis. We found that lot of chemicals which are known and they are listed under the EDC compounds. You can see this beta cytosterol and octadecanoic acid and this diethoxy and this also complex compounds all are actually listed under the EDC. These compounds which are listed under EDC has been shown here. And this reference US EPA has screened such pollutants from the industry as the EDC compounds. This actually analysis I have done from different places of the country and from the different industry, pulp paper as well as kraft paper, both industry, which showed that a lot of the pollutants which are coming in the discharge waste, they are having mixture of the various pollutants which are showing the endocrine disrupting property. So this has been done thoroughly for the analysis part. This established the fact that these pollutants are not known much in India, and for their disposal, no guideline is also still regulated by the regulatory agency. So these are the challenge and these are the problem which has to be disseminated among the people and academicians for the maintaining of the natural resources for the conservation. So these are the important points which I thought to be highlighted through this seminar among the agronomists and farmers and other audience who are directly stakeholder for the natural resources, and among the scientists it has to be uh, highlighted.
Thank you.
This is the report which actually has given a list of the EDC compound, and I found that these pollutants showing the mutagenic and toxicity part, and it has shown the cytotoxic effect as well as chromogenic effect both. So this established that these pollutants are having directly the EDC property or mutagenic property. Seed germination was also affected by these pollutants, which also showed the amylase activity was inhibited. And this finding was published in the Aquatic Toxicology Journal, and the detection of various pollutants of these compounds, which has been shown in front you, was also published. For the detoxification part or the management of these pollutants, we try to see their degradability through biostimulation process, and we optimize this technology in our laboratory, and we found a lot of the indigenous bacteria are available there, autochthonous bacteria which are present inside the effluent itself. If they get the nutrient properly, then they can grow and they can detoxify these compounds uh, before discharging. And these are the just enzyme available in the bacterial community itself, which was detected, and these are just showing the bacterial population during the degradation process, how they are multiplying and growing. So these are some graph or data showing the degradation and detoxification process during the biostimulation, and these are the enzyme correlations during the growth. So due to the limit of the time, I am just going very rapid to highlight the problems and matters which is available here in the uh, topic. So this is just showing the degradation and reduction of the pollutants during this detoxification process. And we have proved that with bacterial degradation during biostimulation process can be an effective technology which can be treated or the effluent at the tertiary stage treatment. So which will be helpful to reduce the pollutants for the protection of the natural resources. It has been shown that after treatment, a lot of the compounds which were present, they were removed, and some different other pollutants as metabolites were generated, and but some were still as the recalcitrant pollutants, which were not gone by this one stage treatment. Then therefore we try to make the bio augmentation process integrated with the biostimulation, and we found that was very much effective. And this biostimulation process technology we could publish in Frontier Microbiology. You can see the view nearby the industry also, the discharge of the pollutants and sludge, how this is affecting to the mustard crop directly. But this crop is very potential, and they show the metal accumulation as well as growth on the sludge directly. Uh, and some actually plants are very much potential for the degradation, or they can grow and they can accumulate some toxic compounds through their root. And this is a Phragmitis communis plant, which is a very much potential. And a lot of the rhizosporic bacterial community I have shown, and I have isolated it. They were found very much potential, and they were facilitating the plant growth for biodegradation and bioaccumulation. So we have also published this finding recently in bioresource technology. And you can imagine and see in the view of the slide also, a lot of the wheat plant and some grass in nature itself showing the potentiality for their recovery of the various metals and pollutants in nature, but they require actually the optimum condition for their accumulation and for this detoxification of the pollutants, which needs proper uh, optimization for the uh, protection of the bioresource conservation. So these are the view You can see this grass is very interesting. Cannabis, sativa, parthenium, even this is a camelina. Mustard is one of the crop which can grow nearby the pollutants and sludge also. And the mustard is well known for the accumulation of cadmium and other metals. So these, but if they are growing the crop plants, they can affect the food quality and the product quality of the plant. So that is very challenging part, and that requires a special attention by farmers and scientists how it can be monitored. So this is the interesting points. I have isolated and estimated some metals taken by the crop plant Triticum brassica. This is the Eucalypta, which is a medicinal plant, Solanum. They accumulated the metal in nature itself, where they are being affected from the effluent and sludge. So this showed that lot of the plants has a potential for accumulation of metal and detoxification of these pollutants, and they can be used for the management of these pollutants as a through phytoremediation technology. But that requires optimization with the proper condition. So this has been already recorded by my team, and it can be used for the control of these pollutants. But since in the industry it is a very high concentration or amount, so that has to be considered into the optimization. This slide showing actually granules of the metals taken by the plant root, uh, how they have taken in the Brassica parthenium and Camelina plant. So this is just showing the potentiality of the plant as a hyperaccumulator, because they have tolerance capacity, and this multinucleolus formation is showing the potentiality of the plant for their adaptation. So there are several plants which has a very high potential for their adaptation at the highly polluted site. This type of finding also I published in Ecological Engineering Journal for the phytoextraction capacity of metal from the pulp paper waste. And this Eclipta and alternanthera has been reported from different places. I have collected this plant and sample from more than 3-4 industry and different part of the country, and found that alternanthera was very much potential uh, for accumulation of different metals and from the sludge directly. So you can see how these discharge effluent is affecting.
Please conclude, Dr. Sabu.
So this is directly coming to our health, and this is just a view of the plant. They are growing on the sludge itself directly, and related with this topic itself, for the sustainable development and bioremediation, I published book from CRC, USA. So I am thankful and acknowledged to different funding agency uh, for the analysis part, for the IITR Lucknow, Baba Sahib Bhimrao Ambedkar University, and for the SAM analysis from AIMS New Delhi for funding DBT and DST. So, and this is my team and research scholar who supported work, and I am thankful to organizer uh, Professor Sanjay Arora for the inviting me as a lead speaker. And since it was very less time, in limited time, I just given a glimpses of my work, how it can be useful for bioresource conservation and management, plant and bacteria both. So thank you very much, sir. I will be pleased to reply any query if there.
Okay, thank you, Ratnam, for a very elaborate and nice presentation. I think it was an eye opener for us, because we agriculturists daily deal with the weeds like alternanthera, you told, Camelina bengalensis you told, Parthenium you told, even Brassica is a major oilseed crop. So
Yeah.
I think they have got their utility in managing these what you call heavy metals and pollutants and these endocrine disrupting compounds. So only thing I would like to request, because I will leave the discussion in the end of the session, I will just like you to request you to, because you just informed us that even the our pollution control board guidelines does not have the mention of those compounds which you have found in your discharges of these pulp paper mills. So if you kindly provide those compounds and their critical limits to ANRCM Secretariat, we can, on your behalf and on the behalf of ANRCM, we can write to UP government or to India government or to Pollution Control Board that these are the compounds which are not yet included in our guidelines or standards, whatever you say, and it should be included while monitoring the effluents from the
That is
pulp paper
true.
mills.
Yeah,
So,
that is true, sir.
yeah,
This is very interesting because globally people are diverting their mind regarding these pollutants which are not so far listed.
yes,
China, Korea, Israel, they have reported well recently. And in India, it has been very few report has been published because this is not known so far and it is affecting to the aquatic resources,
yes, yes.
including the
So,
river water also quality is being affected due to these pollutants.
yeah,
We'll talk about the BOD, COD, and through by any means they control the BOD, COD, or they tell false data. But the ground level situation remains same. This is the reason. And you are very much true through this webinar or seminar, such type of things has to be highlighted in
yeah,
through the
this
thing.
should be the part of our recommendation so that your
Yeah,
findings
that is very important because
that,
I have shown the picture
yes,
which
yes,
is taken by me from the ground itself
yeah,
in India, different parts, and I spoken this problem in front several time in on the channel also TV debate.
yeah,
So this is a high time to take into consideration such type of pollutants for the
yeah,
policy makers as well as for the farmers itself. So
right,
thank you
right.
very much, sir. Further,
Okay.
if there is any questions, I may reply
Yeah,
for
in fact, I expect there will be so many questions, and I
yeah.
think we are not having that much time. So I will request them to just put that questions
Yeah,
in chat
they
box.
can put question on my mail also.
Yeah,
They if
yeah,
they have some time
in
interaction,
your
they
mail
can call me
or in the
also.
chat box of this particular session.
Yeah,
So in the
no
end, we can
problem.
take up those questions and kindly be available if at
Yeah,
all there are many questions. So thank you, Doctor. Thank you so much.
thank you very much. Thank you, sir.
Really it was quite interesting and informative talk. We welcome.
Thank you very
Thank you
much.
so much. So
Thank
we will move on to the next speaker, that is Dr. A.K. Bhattacharjee, and he is from IICAR CISH Rahman Kheda, Lucknow, and he is going to present his talk on in vitro degradation of imidacloprid, that is a insecticide, in sterile and non-sterile soil by microbial consortium. So, Dr. Bhattacharjee, sir, are you available? Can you hear me, Dr. Bhattacharjee? Dr. Bhattacharjee.
Please,
Yeah,
yes, sir.
I can see you, but I cannot hear. So you are not audible.
डॉक्टर भट्टाचार्य जी आवाज नहीं आ रही है.
अपना माइक ऑन कर लीजिए, डॉक्टर भट्टाचार्य जी. And please stick to time, rather 15 minutes, take only 10 minutes, 5 minutes we can keep for discussion.
डॉक्टर भट्टाचार्य जी आवाज नहीं आ रही है. आपका माइक्रोफोन में प्रॉब्लम है.
We cannot, Dr. Bhattacharya, can you hear me? We are unable to listen to your voice, sir.
सर आवाज नहीं आ रही है।
इनको फोन करके बता दीजिए अरोरा साहब।
अब बोलता हूँ।
शायद इनको एहसास नहीं है कि इनकी आवाज नहीं आ रही। अब शायद हमारी भी नहीं जा रही है इनके पास, इसीलिए नहीं होगा।
डॉक्टर अदक, नहीं आ रही डॉक्टर साहब। आपको हमारी आ रही थी?
हेलो, नमस्कार।
Actually, I have talked with Dr. Bhattacharya. There is some issue in his computer, so he is moving to a next computer. Meanwhile, we can have one or two questions from participant for Dr. Professor Chandra if there are. Otherwise, we can go for oral presentation. Meanwhile, he will be ready.
He is not able to connect.
In 7 minutes, he will shift to next computer.
Okay, so let him take 5 minutes. We'll move on to oral presentations. And there are 11 oral presentations listed in this session. May I request Ms. Vandana Kumari? Is she available with us? Vandana Kumari? Not available. Then
No.
Arora sir, who is going to present? There is a paper from you, Dr. YP Singh, AK Singh and Rupam.
Rupam will present. Rupam.
Okay, so Rupam, take only 5 minutes and just tell title, the objective, the findings and the recommendation. And don't take more than 5 minutes, please. All the oral presentations should restrict to 5-minute time limit and just 4 things they should tell. That's all. So please, Rupam, the second paper, that is crop residue decomposition through potential microbe to combat residue burning. Please.
Sir.
Yeah.
Okay. So shall I start, sir?
Yeah, please. Go ahead.
Sir, but presentation.
Who will load? Dr. Aurora, you will load presentation?
I will load.
Please. Yeah, it is visible. Now you go ahead.
Good evening, everyone. My name is Rupam Sahu. Here I'm going to present an oral presentation on the title Crop Residue Decomposition Through Potential Microbes to Combat Residue Burning. Sir, next slide, please. Okay. So Why crop residue burning is in practice? Crop residues are the leftovers on the agriculture field after the crop has been harvested, including stalks and stubbles. And uh, farmers opted for crop residue burning as an easy and inexpensive solution because of the rice wheat cropping pattern, and they need to prepare the field to sow the next crop well in time. So, in order to remove the million tons of residue, they choose burning. So, please next slide. Okay, so why we care about crop residues burning? As farmers opted for it burning as an easy option and inexpensive, then why we care? What is the concern that generates burning? There are some problems burning creates: global warming and loss of plants, nutrients, biodiversity, deteriorate soil fertility, emission of greenhouse gases, loss of soil flora and fauna, release of soot particles causing smog, health hazards to humans, animals, and birds, loss of biodiversity, loss of carbons. So please next slide. So because of these detrimental effects, the maize residues responsible for producing 45.4% air pollutants in the air, and air pollutants, and causes emission of air pollutants, while wheat contribute 25.6% and sugarcane contribute 44.1%, rice 24.9%, and about 50% of crop residues are burnt and released, causes emission of air pollutants by only continent Asia, where Europe produce 11.3% air pollutants through burning of crop residues only. And Oceania contributes 1.5%, Africa responsible for producing 11.5%, America 25.7%, but the number of Asia is very high. Sir, next slide, please. So there are as we can see through the graph, that there are top 10 emitters which causes emission of air pollutants through crop residue burning, and China is the largest. China is the country which which produce highest emission. And the Pakistan, then the India, then United States of America, then USSR, then Brazil, then Indonesia, then Russia, then Mexico, then Thailand. And Pakistan is the least residue burning occur by Pakistan. So next slide, please. So the alternatives are available of burning, like use of agriculture machines such as straw management system, existing combine harvester, happy seeder, rotavator, etc., biochar production, biothermal power plants, residue as fodder for animals, bedding material for animals. We can use these alternatives, but these alternatives also has their demerits. So next slide, please. Okay, so machines favors big landholders, as machine favors big landholders, the small farmers cannot afford such big machines as they are cost ineffective. They require high demand of labor and electricity, and the removal of crop residues for cattle feed also increases nutrient removal from croplands and harms soil with multiple adverse impacts, ranging from soil erosion and degradation of soil to hampering soil, air, and water quality. And in situ incorporation of residues to completely degrade the residues take months to years. So this is also another demerit. It gets difficult for the next crop to grow properly on time. So next slide, please. So why we are approaching microbes? Because microbes are the natural decomposers present in the environment. They break up dead plant, animal biomass and act on the detritus to reform or recycle soil nutrients. Microbes are also responsible for degradation and recycling of elements, basically constitute the foundation of ecosystem. Microbes are efficient and cheap alternatives, as well as they are eco-friendly for the environment. They are eco-friendly and for the degradation of lignocellulosic complex matrix without releasing any toxic compounds to environment that is detrimental to life. So how it works? Microbes decompose the complex aromatic structures of residues by producing lignocellulosic enzymes. The enzymes are biocatalysts involved involved in the breakdown of lignin and cellulose materials into further hydrolyzed useful products. Microorganisms, being in a sexual type of possess fast replication ability. They replicate very fast and they produce enzymes through the enzymatic system. They catalyzes the organic and inorganic compounds naturally, so which can help in the degradation of the lignocellulosic compounds. So next slide, please. Okay, so here is the mechanism. As lignin is a complex amorphous heteropolymer with a three-dimensional structure composed of phenyl propane derivatives linked to each other by irregular coupling of carbon-carbon and carbon-oxygen bonds. It has three structural monomers. H types is para-hydroxyphenyl units, guaiacyl units, renzyl units. So lignin linked to both hemicellulose and cellulose also. So this lignin is solely heterogeneous aromatic compounds. It is very difficult to break this compound. So microbes, especially the white rot fungi, are capable to degrade this complex aromatic compounds, producing enzymes, lignin peroxidase, lacase, manganese-dependent peroxidase, versatile peroxidase, didecyclizing peroxidase, so, and break down the bonds of the complex aromatic compounds and release them into single compounds.
Now you conclude, please. You have taken more time than scheduled.
Okay, sir. So this is the mechanism that producing several enzymes as suitable enzymes.
Come to the conclusion.
Okay, sir. So please next slide, sir. Okay.
Yeah.
As microorganisms are the natural decomposers and possess the efficacy of converting the complex aromatic structure compounds into nutritionally rich compost, such as wood degrading microbes, mainly fungi and bacteria. A diverse spectrum of lignocellulosic microorganisms present in the environment which delignify the various types of plant cell walls. In order to combat the agricultural crop residue burning, we need to find the microbes which show most efficient hydrolyzing enzymatic system. And microbes are definitely, they are capable of producing such enzymatic system in order to degrade the lignocellulosic compound, so that the burning practice of residue can be replaced, which in turn generate harmful consequences by microbial decomposition. So we can definitely opt for microbial decomposition of crop residue as an alternative other than burning. Okay, thank you, sir.
Thank you for a nice presentation and a very clear-cut presentation. Thank you. Dr. Bhattacharya, are you available now? Can we take your presentation, sir? Dr. A.K. Bhattacharya? No response. So now I move on to Mr. S.K. Yadav. S.K. Yadav जी available हैं? नहीं available है. The next presentation is Supriya Vyas.
ये आज नहीं है अभी.
Pardon, sir?
ये सुप्रिया वैस नहीं है, ये कल करेंगे अपना.
नहीं है. Then नीलिमा गर्ग मैडम. Dr. Nilima Garg, A.K. Bhattacharya and Pradeep Kumar Shukla.
Dr. Bhattacharya जी आ गए हैं.
Yes,
okay.
Sir, ready.
Okay, that's
Voice
nice.
is audible now?
Yeah, now you are audible.
Okay, thank you, sir.
So, degradation of imidacloprid. We have a talk on this.
Good afternoon, everybody.
Yeah.
I am going to present our investigations on in vitro degradation of imidacloprid in sterile and non-sterile soil by microbial consortium. This is the chemical structure of imidacloprid, whose IUPAC name is 1,6-chloro-3-pyridinyl methyl N-nitro-2-imidazolidinimine, and its chemical formula is C9H10Cl N5O2, having a molecular weight of 255.66. Imidacloprid is the first neonicotinoid insecticides invented and marketed by Bayer at 1992. It is the most widely, or you can say extensively used insecticide among the neonicotinoid groups against soil insects, sucking insects, termites, and some biting insects in various crops. including rice, cotton, cereals, maize, sugar beet, vegetables, and fruits like grapes, mango, citrus, etc. However, it's a persistent insecticide in soil, and its residue can persist for more than 1 year, depending on soil type or soil environment, with half-life values ranging from 29 to 1230 days. It's so persistent. It's around 3.5 years. This scenario warrants eco-friendly and cost-effective degradation of imidacloprid, and natural microbial transformation seems to be an effective and viable approach. Biological removal of chemical pollutants becomes the method of choice nowadays, because microbes can use a variety of xenobiotic compounds, including pesticides, for their food source, and in the process, they can mineralize and detoxify these chemicals. Several microorganisms are isolated and identified from contaminated soil, which are capable of degrading imidacloprid. These include Lipsonia species, Pseudomonas species, Bacillus species, Acrovectum species, Enterobacter species, Burkholderia species, and so on. However, very few studies have been conducted on simultaneous microbial biotransformation of imidacloprid in sterile as well as non-sterile soil, and that too using microbial consortia. Therefore, the objective of the present study is to isolate defined microbes from imidacloprid contaminated mango orchard soil and their capability as consortium to degrade imidacloprid in both sterile and non-sterile soil under laboratory conditions. Soil samples are collected from mango orchard located at Ramankela, Lucknow, that is at our institute, ICICISH. Isolation and purification of culture having imidacloprid degrading potential was done on carbohydrate utilization broth and agar with 1% imidacloprid formulation, that's media, 17.8% SL, suspension liquid. Imidacloprid was used as the carbon source, and identification of microbes was done using 16S rDNA gene sequencing technique. 10-gram soil samples, the sampling was done using the quartering method, where in petri plates were treated with two different concentrations of imidacloprid, that's 5 and 10 ppm, maintained in triplicate and kept at room temperature up to 50 days, along with control. Control, there is no microbes, but imidacloprid is there. And the extraction of imidacloprid from soil was done using acetonitrile, and the pesticide was estimated using HPLC having C18 column and acetonitrile water 3565 as whiteage. Based on their plant growth promoting and enzymatic activities, 2 bacteria and 3 fungi were identified. Actually, we have isolated 16 microbes, the 4 fungi and 12 bacteria. But the identification has been done for those bacteria and fungi who have shown promising plant growth promoting activities and enzymatic activities. Those 2 bacteria were Pseudomonas moseli strain NG1 and Spingobacterium mucilaginosum strain NG201, whereas 3 fungi were identified as Aspergillus oryzae strain NG203, Aspergillus acuminatum strain NG202, and Penicillium oxalatum strain 201. In non-sterile soil, it has been found that imidacloprid degradation by microbial consortium was at par in both lower and higher doses. Almost at par, 52.63 and 51.85, there is almost no difference for 5 and 10 ppm concentration after 50 days of inoculation. Similar results are obtained from sterile soil also. But here the degradation is higher as compared to non-sterile soil. 72.69 and 73.21% for 5 and 10 ppm concentrations. However, compared to control soil, which is the degradation of imidacloprid was significantly better in both treated and sterile and non-sterile soil. In case of non-sterile soil, 43.21 and 41.01% degraded from 5 and 10 ppm concentrations, and in case of sterile soil, 51.7 and 53.7% of imidacloprid was degraded from non-sterile and sterile soil. And it has been found that the degradation of imidacloprid followed first order rate kinetics in all the soil samples, whether it is sterile or non-sterile, or with or without microbes. This is the table from non-sterile soil. This is from sterile soil. So, from the present study, we can conclude that microbial consortium could be used effectively for degrading imidacloprid in both sterile and non-sterile soil under laboratory conditions. Thank you.
Thank you, Dr. Bhattacharya, for a sweet and short presentation. It was very crisp, and I think everybody understood it well. So I think if there are any queries, kindly put that in chat box, and Dr. Bhattacharya will be able to answer those queries in the end of this session. Thank you, sir. Thanks a lot. It was a very nice presentation from you. Let us move on to the next presentation. And who is next? S.K. Yadav, are you available? Resource conservation practices under organic farming? Not available. Supriya Vyas, fungal community diversity? Not available. Dr. Nilima Garg and then Dr. Bhattacharya and Pradeep Shukla, metagenomic analysis of imidacloprid rated mango orchard soil. Similar kind of presentation, sir. Somebody is going to present, or who will
under
present?
pre-condition only, Pseudomonas mosalis 10 NG21 has been studied.
Okay, so no need for presentation, huh? Then Ratna Sahai, I think she is not available as told by...
She is there, Ratna is there.
Ratna is there, okay. So impact of halophilic bioformulations with different levels of NPK on wheat crop and fertility of sodic soil by Ratna Sahai, A.K. Singh, S. Arora, Archana Singh, R.C. Moria, D.K. Tiwari and Sunil Singh. Please.
A very good evening to all of you, sir.
Yeah, we can.
Thank you, sir.
Yes. So here I'm going to present my presentation on impact of halophilic bioformulation in sodic soil on wheat crop and its fertility level. So the basic objective of my study is to, as I am working in KVK, Krishi Vigyan Kendra, we are dealing with farmers. The basic, my objective is to encourage the farmers to use the biofertilizer and reduce the excessive use of chemical fertilizer, and to improve the natural condition of soil there, to improve the quantity and quality of the produce. And last but not the least, most important factor that we are trying to get is to reduce the cost of production. Here is my experimental information. Sir, we have, we have about 21,386 hectare area is highly salt affected sodic soil in Unnao region. That's why we have selected this study. We have taken a village, Munshi Kheda, where the farmer's field, which we have selected, the soil condition was, the pH was about 8.9, and in previous condition, they were using the local variety without using the biofertilizer. What we did, we have been included the salt tolerant variety, that is Kerala 210, with the halophilic biofertilizers, Halo Azo and Halo PSB, and we have taken the 4 treatments, control and with Halo Azo, with Halo PSB, and the 4th one with the both of them. We have included in on-farm testing, and in our this experiment, what we have recorded in very early stage, we got that the inoculation of biofertilizers improved the germination, and we have reported that about after inoculation of these biofertilizers in all of these 3 treatments, it promotes the early germination of seeds. 3 days early emergence was reported in all the inoculated treatments as compared to control. Other than the early germination, the germination percentage was also recorded higher, as in case of Halo Azo, 7%, PSB 9%, and in both of them, 12% higher germination was recorded as compared to control. Other than the germination, if we come to our production, about 10% more grains per spikes were recorded, and due to higher grains per spikes and grain weight per spike, about 10% higher grain yield and 11% higher straw yield was recorded at farmers' field. If we come on the effect of bioformulation on nutrient content, here we can see the both of the bioformulation were able to improve the nutrient content in the plants individually and in co-inoculation. Phosphorus content of the plant increased higher in case of PSB, and if we include both of the bioformulation, then here we can see the better result as compared to sole inoculation of the biofertilizers. If we see the properties of the soil, in post-harvest soil, it showed the pH was lower and organic carbon content was higher in the bioformulation treated soil. Although we are doing this experiment from last 2 years, even then we have recorded about 0.2 lower pH as compared to control. Other than the pH, the organic carbon content of the soil was about 0.32% as compared to control 0.22, and here we can see that the if we are inoculating both of the bioformulation together, then they are improving the nitrogen as well as phosphorus content of the soil. Other than the these nutrients, the micronutrient level was also slightly improved. If we see the enzyme activity, dehydrogenase activity, it was recorded up to 25.5% higher as compared to control in the inoculation of both the bioformulations. Now here's the outcome of the study, that the cultivation of potential salt-tolerant variety with the halophilic bioformulation in salt-affected soil is the best way to improve the crop productivity, and it can play an important role in limiting the excessive use of chemical fertilizer, which we are facing right now in farmers' field. They are using excessive fertilizer without knowing the dose or without knowing the biofertilizer means. So we are impressing on that, and the results are really impressing, and farmers are getting impressed with our experiments. Thank you, sir.
Okay, thank you. I just have one query. First thing that you inform the audience, what is halophilic?
Yes, sir. These are the bioformulation which are active at high pH in high salt conditions.
Okay. Second thing, it was seed inoculation.
Sir, we are using in wheat seed inoculation and in soil inoculation also, just to improve the effectiveness.
Okay. Thank you so much for a very nice presentation. Thank you. Thank you. So, who is next? Dr. S.K. Verma and O.P. Rao. Effect of fertilizers on turmeric and guava yield under Agri-Sylvi Horti system on partially improved sodic soil. Ratna Sai, your presentation was nice. It is over. Thank you. S.K. Verma साहब हैं, अवेलेबल हैं? नहीं है. Then let us move on to Mr. Sandeep Kumar, Thomas Abraham, Manoj Kumar, Lipi Reena and Nikam Kumari. Anybody from this team for the presentation on effect of different cultivars with fertility management through organic manure on growth and yield of wheat? Anybody from Sandeep Kumar, Thomas Abraham, Manoj Kumar, Lipi Reena and Nikam Kumari? कोई नहीं है. Then moving to Varsha Pandey and Ajay Srivastav. Are you available?
Yes, sir. Sir,
Okay.
Varsha Pandey.
Good. Effect of integrated use of chemical fertilizers, FYM and green manure on soil fertility after maize harvesting in maize wheat cropping system in a molly soil. So we are going to hear presentation from Varsha Pandey. You are welcome. Please go ahead.
So just a minute.
Yeah.
So is my presentation visible?
Yeah, it is visible.
Okay, sir. Good evening to one and all. I am Varsha Pandey. I am PhD research scholar from GB Pant University of Agriculture and Technology, Panagar. Today I am here to present on the topic effect of integrated use of chemical fertilizers, farmyard manure and green manure on soil fertility after maize harvesting in maize wheat cropping system. First is introduction. We know that agriculture plays a vital role in India's economy, and the rate at which Indian population is increasing, it has pressurized Indian agriculture to produce more from the shrinking arable land. And therefore, it is very important to pay attention to the maintenance and to the improvement of soil fertility. And organic materials, they hold a great promise as they are a source of multiple nutrients because of their ability to improve the soil characteristics. Therefore, integrated use of the inorganic fertilizers along with the organic manures, it can help us. Therefore, the present study has been undertaken and the study area was a crop research center at GBPOT Pannagar, Uttarakhand to study the integrated use of inorganic fertilizers and different manures. These are the treatment details. The experiment was laid out in RBD with 9 treatments and 3 replications. Dhecha was the green manure which was used and it was sown during April month, and a 45 days old dhecha was then incorporated into the various plots. These treatment combinations, it consists of recommended doses of fertilizers which are recommended to the farmers. Then it consists of use of inorganic fertilizers, and these inorganic fertilizers were applied according to STCR doses. STCR is soil test crop response. STCR equations are developed using a statistical method, and then various fertilizers are applied to the field based on the soil test value. If the soil has a higher amount of a particular nutrient, then using the STCR equations, less amount of that particular nutrient is applied. So it helps in maintaining a balanced application of nutrients. Now, these are the results. The application, it was seen that the application of fertilizers, farmyard manure, and green manures significantly increased the water holding capacity of the soil over control. Maximum water holding capacity was seen in case of a T4 treatment, where fertilizers as well as farmyard manure was applied. And the increase could be attributed to better root growth and more plant residue addition under these treatment. And also there was improvement in the soil aggregation in the T4 treatment. Then if we talk about organic carbon, the highest organic carbon percent was observed in case of T5 treatment, where only 75% of the dose of chemical fertilizers were used, and 25% of the fertilizer doses were substituted by the green manures. And the organic carbon obtained in this treatment was at par with T4 treatment, where integrated use of fertilizers and farmyard manure was done. And this might be due to enhanced root growth, which resulted in more organic residue in the soil, which after decomposition might have increased the soil organic carbon content. Then if we talk about the primary nutrients, the higher amount of NPK was observed in case of T4 and T5 treatments. The increase in the available nitrogen content, it might be due to the application of the manures, which attributed to more mineralization of nitrogen and greater multiplication of the microorganisms, which converted organically bound nitrogen into inorganic form. And lowest amount of NPK was seen in case of the control. That can be because of more mining of the nutrients, and there was no fertilizer addition in the control treatment. Then if we talk about phosphorus, the buildup of available phosphorus with the combined use of fertilizers and organic manures is due to the release of organic acids, which help in more decomposition of the nutrients, which help in releasing more phosphorus, and also organic matter decrease the phosphorus fixing capacity. So we can conclude from the study that imbalanced and excessive use of chemical fertilizer has led to a significant degradation of the soil health. Thus, inorganic fertilizers along with organic manures is a viable option for restoring the soil organic carbon and nutrient turnover, thereby improving the availability of nutrients in soil and maintaining soil quality for a very long term. Thank you.
Okay, thank you, Dr. Varsha. Your presentation was very nice and crisp. One thing I would like to know for the audience that do you have a STCR equation for maize at Pantnagar?
Yes, sir.
Okay, good. So finally, your conclusion is that INM along with green manuring is good for maize.
Yes,
as well as for maintenance of soil health.
Soil fertility. Yes, sir.
Okay. Thank you so much. So it was nice presentation.
Okay.
And now let us move to next presentation, that is weed management in medicinal and aromatic crops under organic farming. And the people who are going to present, they are Priyanka Suryavanshi, Dipender Kumar, Pooja Maurya, Abdul Majid, and Sri Ram Rajvanshi. Is there anybody among the authors available for presentation? Priyanka Suryavanshi, Dipender Kumar, Pooja Maurya, Abdul Majid, and Sri Ram Rajvanshi. कोई है? कोई नहीं है. Then let us move to the next, and that is the last oral presentation for this session. Anita Jaswal, are you there?
Yes, sir.
Okay, good, nice. Carbon sequestration by biochar for sustainable soil and environment. Good presentation, I think it will be. Anita, please.
Yes, sir. Very good evening to all of you. My name is Anita Jaswal. I am from LPU. I am a PhD scholar. I'm just going to present my views on carbon sequestration. It is a journal information. I just like a case study. So carbon sequestration by biochar for sustainable soil and environment. This is my topic. So today we are facing the recent issue regarding the global warming. So for that global warming, the main gas is responsible is carbon dioxide. So carbon is the most abundant element in the living things, and it approximately accounts for 50% of total mass of the plants and the animals. So carbon cycle is directly very closely related to the global climate change, because due to the emission of the carbon dioxide due to the human activities like burning of the straw, like or originating from the fossil fuels or the land use change. So atmospheric concentration of carbon dioxide has increased from 280 ppm from pre-industrial era to 385 ppm in 2018. That is approximately 37.5% increase. And in present, it's increasing at the rate of 2 ppm per year. So there exists a strong link between the food insecurity and the climate change. As we already know that part, our agriculture is totally based upon the climate. So climate plays a very vital role in the crop production part. So if there is we are facing in the issue regarding the climate, obviously the crop production is going to be affected. So these two are the major crises: climate change and food insecurity, that can be addressed by restoring the soil organic carbon pool through the carbon sequestration in ecosystems, more specifically in the agricultural soils. So first of all, I want to discuss what do you mean by carbon sequestration. It is the process involved in the carbon capture for the long-term storage of the atmospheric carbon or the other forms of carbon, so that we can mitigate the global warming part. So after that, the second part is of my topic is to maintain the soil health also. So soil health is the foundation of the vigorous and sustainable food system. So as we are continuously cultivating the land, so we are going to disturb the natural system like nutrient cycling, release and uptake of the nutrients. So it's a very important to maintain the threshold level of the soil, so that we can maintain physical, chemical and biological properties of the soil and maintain the sustainability for the production of the crops. So global potential of carbon sequestration in soil of agro ecosystem is about 2.1 billion tons carbon per year. So if we are going to sequester that atmospheric carbon in the soil, then it's going to increase the soil carbon pools, which reduces the atmospheric concentration of carbon dioxide. Because a part of the photosynthesis, because there is a cycling between the atmospheric carbon and the plant cycle. So there is a cycling of the carbon, so that plants are going to utilize that atmospheric carbon dioxide in the photosynthesis process, in which that biomass is converted into a humus and it's going to resist in the soil for a long period of time. So this is the way of the carbon cycling, how the plant going to absorb the carbon dioxide from the atmosphere. And it's going to first it absorb the carbon dioxide from the atmosphere. It's going to utilize that carbon dioxide from the air and water to build the for using in the process of photosynthesis. And then plants exudate that carbon through their roots to feed the soil organisms. And after that, the soil organism release the carbon dioxide through the respiration process. Again, that plant's going to absorb. So in this way, the cycle is going to be completed. So after that, the main issue is that in India and most probably in Punjab, the major issue is the stubble burning or the straw burning. So availability of the biomass in India is approximately 500 million tons per year. So these residues either we can partially utilized or unutilized due to the various problems. So out of this 500 million tons, 93 million tons of the crop residue burn each year in India. So residue burning is a very important part. According to that, it's a fast, according to mentality setup like that, it's a fast way to clear the agriculture field. But along with the loss of the valuable biomass, there is also the loss of the nutrients. Along with that loss of the nutrients, there is also a release of the toxic gases like greenhouse gases. So there is a very need to efficiently use of this biomass available as a crop residue and other farm waste, which is converting into a useful source as a soil amendment. So number of options are there for carbon sequestration and the increase the fertility status of the soil. But the most modern approach in today is that application of the biochar. That is the charcoal created by the low temperature pyrolysis of biomass under the anaerobic conditions. So this is just a brief description of the biochar. So it is a biochar is nothing. We are going to heat the straw at a particular temperature, most probably under the pyrolysis condition. Pyrolysis condition means in the absence of the oxygen or anaerobic conditions at a certain temperature, and most probably we are using that temperature 250 to 600 degrees Celsius. So that is totally based upon the raw material, whatever we are using for the feedstock and the raw material. So it is a recalcitrant in nature and rich in the carbon. So when we are adding this particular biochar in the soil along with the fertility status, it also mitigate the helps in the mitigation of the climate. So this is just a flow diagram. So this is one is the plant, biomass is there. So we are going to pyrolysis at a certain temperature. So from after the pyrolysis, first thing is made, that is a biochar. So that is we are adding it to the soil and it is again returned to the soil, 50% of that. During this process, biofuel is also going to be prepared, like bio oil or hydrogen that we are using as a syngas in the transportation or transport or energy, like that part. So this is just so many other uses are also there for biochar. So this is a just finding biochar impact on soil properties. So physical, chemical and biological properties are going to be improved with the help of the biochar. If I am just discussing about the physical properties, so first very important part is the bulk density. So when we are adding biochar to the soil, it reduces the bulk density of the soil and increases the porosity. If porosity is increasing, the water holding capacity and the nutrient retention capacity of the soil is also going to be increased. Along with that, the leaching is also going to be decreased. So as you can observe from this, so different properties are there, like cation exchange capacity, fertilizer use efficiency, crop productivity, so bulk density, methane emission. So this is a general finding. So percentage, how it's going to be increased by addition of the biochar. So the main part is how the biochar helps in the carbon sequestration. So declining the fertility status of the cultivated lands, the fertility status of the soil is going to be down day by day, specifically due to the reduction in the content of soil organic carbon. Most probably in Asian and African countries, which also responsible for the low crop production and the resource use efficiency. So soil organic carbon is a major part that affected the soil quality. So it is an important indicator of agriculture sustainability. So restoring soil organic carbon is important not only to achieve the food security, but it also improves the quality of the soil. It increases the NPK content of the soil also. Along with that, it also plays a very important to change the active pools of the carbon to the passive pools, because when we are adding the biochar to the soil, it's going to change the nature that aliphatic, during the process of the pyrolysis, aliphatic carbon is going to be changed into the aromatic carbon. That is going to, the carbon is become recalcitrant in nature and persist in the soil for a longer period of time. So it also improves the quality of the soil, and also a main impact to, it reduces the negative impact of the agriculture related climate change. So it has been recognized one of the important strategy to counterbalance the impact of climate change through the sequestering the carbon dioxide into the soil, so that the load of excessive atmospheric carbon dioxide will going to be reduced. So it is a just finding of one paper that earth soil is going to be stored around 4 times more organic carbon than in the atmosphere. So annual uptake, that point I already discussed. If the plant is going to utilize that carbon dioxide during the process of the photosynthesis, that is around 8 times greater than 2 days anthropogenic carbon dioxide emission in the atmosphere. So it is a clear-cut evidence that huge amount of carbon are cycling between the atmosphere and the plants annually, and soil is the one of the most important source where most of the world's organic carbon already stored in the soil. So if we are taking this initiative, then by diverting 1% of the annual net plant uptake into biochar would mitigate approximately 10% in the current anthropogenic carbon emission in the atmosphere. So if we are going to divert a very small proportion of this amount of cyclic carbon into the soil with the help of the biochar cycle, it would make a large difference in the atmospheric carbon dioxide concentration, but a very small difference to the global soil carbon storage. So this is a just cycling, systematic cycle of biochar in soil and plant system. So that first sunlight is there, that plant's going to absorb that photosynthesis process, biomass obtained. Then we are going to pyrolysis process is there. So biochar is prepared during the heating process and volatile compounds also prepared. Then we are adding that part carbon and nutrients into the soil. So that carbon rich product back into the soil and the microbes use. That's the full cycle of that. So this is the overall scenario. Biochar not only helps in the mitigation, but in each and every part, like helps in mitigation of climate change. Agriculture production is going to be increased. We can use the residues in a better way. Helps in the water conservation also because it increases the water retention capacity of the soil due to the more porosity. So soil fertility enhancement there helps in the sustainability and clean energy production is there. So this is a just suggestion from the different findings are there. So it's a not recently it's not a widely concept means not adopted by the farmers at a large scale. So it is a need of today. We have to all of us we have to share this information with the farmers so that we can utilize the residual portion in an efficient manner so that the farmers are going to increase the production. So thank you all of you. That's all.
Okay, thank you, Anita ji. I think it was a nice presentation, and your emphasis on carbon sequestration through biochar really
Yes, sir.
needed. Only thing is that the pyrolysis facility at farmers level that has
Yes,
to be
sir.
created with government support or with any other support. Then
Yes,
only
sir.
we'll be able to realize this dream. Thank you so much.
Thank you, sir.
So thank you. All the presentations were quite nice, and in this session we had two lead lectures. One is on your paper of this pulp mill extract effluents and their what you call polluting effect and their standards and what needs to be revised and how policymakers can incorporate the findings of Professor Ramchandra for updating their effluent standards to check the pollution of our water bodies as well as our soil. Then we had a presentation from Bhattacharji, Dr. Bhattacharji, about the biodegradation of imidacloprid, a very widely used pest insecticide, and it's a very effective insecticide, in fact. So it is very widely used, and we need to have some biodegrading microbes so that we can check the persistence of the particular herbicide in our soils. Then we had four oral presentations, and it ranged from biochar, means this carbon sequestration through biochar in our soil, and then we had some presentation on your this what you call organic integrated nutrient management in maize by using STCR equations and then integrating it with the green manuring. Then we had some what you call biodegradation and something. So I think the session was very much pertinent, and we included presentations right from effluent management to biochar to climate change to microbial degradation of pesticides, and the whole gamut of this bioresource management was addressed, though the presentations were few, and most of the oral presentations and the participants were I think they just could not wait for 1 year, and somehow they just missed their presentations. But I think we take those presentations as read, and we include that in our abstract book. And now I request our co-chairman, Dr. Khare, if she has something to say on these particular presentations. And then at the end, we will have 10 minutes discussion if there is some questions from the very first presentation to the last presentation. So Dr. Khare, please, if you want to say something.
Thank you, sir. Very nice presentation. And this session is very informative. We get lots of knowledge, and in fact, I am also working on the biochar, so I'm pleased to hear this presentation. Thank you again, sir.
Okay, thank you. And I will request our conveners or coordinators that please record the minutes and make 2 or 3 recommendations, not more than 3 from the session, so that that can be included in the list of recommendations at the end of this particular webinar. And any queries from the related to different presentations in the session? We can take up 4 or 5 queries. I can see Professor Ramchandra is available. I can see even other people who presented their findings. So no queries. So I thank all the audience, all the organizers, and the participants for being patient. And at the end, I think we have 20 participants still taking part in these presentations, that too by 5 p.m. So it's a really healthy sign, because even in offline presentations, sometimes we find not more than 5 or 8 participants in our auditoriums. So this is a good thing that we have at least 20 participants. So over to Dr. Aurora for further proceedings. I think I should close this session here. Thank you so much. Thanks a lot. Namaskar.
Thank you.
Thank
Thank
you.
you, sir.
Thank you, Dr. Srivastava, sir.
Thank you.
Dr. Pooja Khare, madam, for sparing your time, and Dr. Ratna Sai and all the other members of the this team. I am thankful to Professor Ramchandra for sparing his valuable time and deliberating on the very important issue. Dr.
Thank
A.K.
you very much.
Thank you, sir. Dr. Bhattacharji, who is very dynamic researcher and the lead speaker of the today's session, and all the other presentations. Thank you very much for all the authors. So we are moving ahead for the poster presentations. So already we have the number of poster presentation listed in this program, but I think only few are available. So we will be taking the one who have already submitted their posters. Then we will allow others also who have with them their posters, they can share also. Dr. Kohli is the chairman of the poster section. So I think he is available, and I will be coordinating from here. First one is P.K. Upadhyay. P.K. Upadhyay, are you ready with your poster?
Yes, sir. Hello.
Yes, yes.
Yes, sir. I'm ready.
You have sent your poster, or
I have already sent my poster.
Okay. Just we will share.
May I share with my mobile?
Mobile, it will be difficult.
Sir, I have no laptop.
No problem. We will, if you have sent us, then we will share.
Okay, sir. I have already sent.
You have sent it today or yesterday?
15.
Okay. Can you see?
Yes, sir.
Start, please.
Thank you, sir. I'm Prashant Upadhyay, research scholar, Mahatma Gandhi Gramodaya University, Chitrakoot. This investigation was done from the years 2018-19. The topic is a study on soil fertility status of Amla planted area in the district of Pratapgarh of Uttar Pradesh. Amla is commonly known as the gooseberry, Umbellica officinalis. Hello. Hello. Are you hearing, sir?
Yes, yes.
Pratapgarh is a leading. Pratapgarh is a leading. Hello. Pratapgarh is a leading Amla production. Leads in Amla production. Production in 384,000 metric tons in Pratapgarh region. The methodology is there was 70 blocks in the district Pratapgarh. Hello. There are 17 blocks in the district of Pratapgarh. The study was done in only 5 blocks where the Amla plant areas, Amla orchard are defined. These blocks are Sadar, Sangipur, Patti, Belkharnath, and Chandwa Chandika. The representative soil profile in each village were exposed. At each location, 2 to 4 composite samples, soil samples were taken from the different versions 0 to 20, 20 to 40, 40 to 60, and 60 to 80, and 80 to 100 centimeters. You can see in the sampling blocks in left side blocks and the name of the village where the sample were digged. Sangipur, in Sangipur block, Atheha, Muraini, and Rahatikar. Chandwa Chandika, Adharpur, Bhadoshi, and the Taranpur. In Sadar block, Bhupia Mau, Bhualpur, and the Gonde. In Patti, Bibiapur, Dubauli, Parsan, and Kukuar. And the Baba Belkharnath, Amerpur, Gopalpur. Methodology of the soil analysis was soil pH, EC, and organic carbon, and PKS, and micronutrients like iron, magnesium, iron, boron, manganese, zinc, and copper. You can see the method by which this analysis was done. The result was of the pH shown in the graph. Its pH ranges from 6.9 to 9.5, and the EC shows in the graph that it was 0.11 to 1.8 ranges of 15 block, 15 profiles. The status of the major nutrients and macronutrients it shows in the graph, right? NPK and the NPK and zinc, manganese, copper, and boron. The conclusion is that the fertility status of the Amla planted soil was range pH I have already said, and the organic carbon. Hello. Hello.
Yes, you carry on.
Mr. Raghavendra, please mute yourself, otherwise we have to remove.
Amla planted area soil in the Pratapgarh that available major nutrient NPK and S content of the soil range from the 100 to 373 kg per hectare, and 4.1 to 57 kg per hectare, 69 to 418 kg per hectare, and 0.47 to 26 milligram kg per hectare respectively, which revealed that the decreasing trend with the depth and the soil were found to be the low to the high amount of major nutrients. Available micronutrients with boron, zinc, iron, manganese, and copper were adequate to high. You can see example like 0 to 5 ranges. It ranged the 0.5 to 0.9. 0.4 to 0.6 ppm, 2.7 to 5.7, 2.1 to 4.99, and 0.84 to 21.8 milligram kg per hectare respectively. The very well availability of the micronutrients shown in all those soil samples. Thank you, sir.
Okay. Okay, Mr. Upadhyay, can you hear me?
Yes. Hello.
Hello?
Yes,
Yeah,
sir.
Mr. Upadhyay.
Ji,
I
sir.
would just like to know whether you have any concluded anything, or you have got any inputs about the suitability of these soils for any other crop. Because amla is maybe a crop which can be grown even on the marginal lands. But what inputs do you want to derive with the study? For
Hello.
maybe a use, for maybe something like for a better use of the land, an alternative land use or something like that.
My study was done only for the those soils where the amla orchard was found in the district of Pratapgarh, sir. I was trying to take the sample from those areas where the amla grown and the productivity.
I understand that you've done the study from amla growing only, from amla orchards only. But then
Yes,
what next? What importance is the study for? I mean,
hello.
बंद नहीं हो सकती देखो.
हेलो, सॉरी सर, आवाज नहीं आ रही है.
चलिए कोई बात नहीं.
I just wanted to ask whether you have got inputs. Do you have?
No, sir, I didn't provide any input.
आप प्रशांत जी सुन लीजिए एक बार आराम से, फिर उसके बाद बताइए.
जी सर.
बोलो डॉक्टर कोहली.
हेलो सर, सुन पा रहे हैं मुझे?
कुछ प्रॉब्लम है डॉक्टर कोहली के पास, ऐसा है
सर. हम भी ट्रेवल कर रहे हैं सर, इसलिए सर मेरे पास भी आवाज कट रही है.
आज तो ट्रैवलिंग डे हो गया सबके लिए, सभी लोग ट्रैवल ही कर रहे हैं.
नहीं सर, वो क्या था ना सर, 5 बजे ऑफिस बंद हुआ तो निकलना ही पड़ा सर, रास्ते में इसलिए थे.
शार्प टाइम में आप. चलिए ये कोई बात नहीं, मतलब कहने का मतलब ये है कि ये डॉक्टर कोहली का क्वेश्चन ये है कि
जी सर.
चलिए ठीक है, थैंक यू डॉक्टर उपाध्याय.
बोलिए सर, सुनाई नहीं दिया, फिर से बोलिए सर. कोहली सर का क्या क्वेश्चन था? जी सर.
ये सिर्फ आंवला के लिए ही है या और कोई क्रॉप के लिए भी यही चीज यूज कर सकते हैं?
ये सर अभी मैंने जो किया है ये तो आंवले के लिए किया है सर, वहाँ का देखने के लिए कि ये देखने के लिए कि रेंजेस क्या है इसका वहाँ का, और आंवला का चूंकि वो ज्यादातर यहाँ पे काटे जा रहे हैं पौधे, तो और प्रोडक्टिविटी लो होती जा रही है, तो ये देखने के लिए कि क्या कारण है इसका जो है फर्टिलिटी स्टेटस चेक करना चाह रहे थे सर. हेलो.
फर्टिलिटी स्टेटस में फिर क्या आपको पता चला, क्या निकल के आया?
ये देखा सर कि ऑर्गेनिक कार्बन तो हेलो.
हाँ, ऑर्गेनिक कार्बन का स्टेटस.
ऑर्गेनिक कार्बन स्टेटस जो था सर ज्यादातर लो कंडीशन में था. रेंज जो आप प्रोफाइल के लिए थी उसमें किसी 1-2 जगह सर 0.6 पाया गया है, और जैसे पीएच यहाँ का जहां भी आंवला लगाया गया है ज्यादातर हाई पीएच पाया गया है कि 9.8.5 के ऊपर ही सॉइल है यहाँ का. हेलो.
तो 9.8 वाली
9.5, 9.1, 9.2, 8.7. हेलो.
हाई पीएच की सॉइल का रिलेशन निकाल लेते हैं ना अगर आप.
जी सर. हेलो.
आप बोलिए डॉक्टर कोहली, अगर आवाज हो पा रही है तो ठीक है, नहीं तो थोड़ा प्रॉब्लम आती है. चलिए वो चैट बॉक्स में आपको क्वेरी होगी कोई तो, है ना? ठीक है.
हेलो, सुन पा रहे हैं सर मुझे?
बस ठीक है उपाध्याय जी, अगर कोई क्वेरी होगी डॉक्टर आपकी तो चैट बॉक्स में डालेंगे. ठीक.
ओके सर.
ओके, थैंक
यू सर.
Now let's move to next presentation. Okay.
next presenter for in this session, Mamta Prajapati, Hiren Das. Hiren Das,
Hello, sir. Hirendas is here.
where is your poster you have with you?
Yes, sir. I have sent to mail already.
Okay, okay. Today or earlier?
Sir, earlier, sir.
Okay, just I am sharing.
Oh, fine.
Is this your poster?
Yes, sir. This is. Okay. Good evening, everyone. I am Hirendas, MS student from Central Agriculture University. Now I'm going to present on changes of soil organic carbon and different mulching practices in citrus orchards of hilly region. Generally, in hilly region, the basic problem during rainy season is soil loss of soil organic carbon with the soil runoff because heavy rain, because the slope will be more because the soil organic carbon is generally lost by the water runoff. So to prevent that runoff, so what are the uh, soil management practices we need to do is one of the best management practices soil mulching. So with the soil mulching practices, there are other benefits also like moisture conservation will be there, some weed control also will be there. So we'll see what are the different type of mulching practices and its impact in soil organic carbon. If we see in the graph, we can see we have taken here, first one is control, second is straw mulching and grass. In this over a overall year, if we observe by three different type of mulching practices, here we got grass mulching has the highest amount of organic carbon found. Means the soil under this grass mulching has found the highest amount of organic carbon. So, and then next one, if we see, and mulching with in the second graph also, like in different depths of soil also, we have found like it's up to 0 to 20 and 20 to 40 in both the depths of soil also. We got highest amount of soil organic carbon found in grass mulching. And here also, if we see mulching and soil organic carbon, here, here it is grass mulching. This is GT shown the highest amount, like 8 gram per kg of soil. It has been tested more than it's 8 grams per kg of soil. It is shown under grass mulch. So it is one of the best mulching practices that can be adapted farmers who are cultivating their crops in hilly regions. And so there are some treatments we can use like different types of treatments has been shown like with comparison to different years also. It is shown farmyard manure shows the highest amount of organic carbon, means it adds more amount of organic carbon with reference to the others one. So it is also one of the treatment that can be adapted, followed in the hilly region. So finally, I can conclude that by following grass treatment and by using farmyard manure, the organic soil organic carbon can be improved, where it is a source of soil soil biodiversity, which increases nutrient availability, and it usually maintains the healthy soil and maintains the crop healthy. Thank you, sir. Hello. Hello.
Oh, okay, okay, Hiran.
Sir.
Have you analyzed these materials like rice husk, sawdust, chopped banana leaves, FIM.
So
forest-
actually, this is my review topic, sir.
Okay. You have taken from other sources. Okay.
I have taken other sources.
Any query from participants? You are doing MSc or PhD?
Sir, MSc, sir, MSc.
MSc in which subject?
Sir, in soil science.
Okay. Thank you. Thank you for your.
Can I ask and suggest?
Yes, sir.
Yeah, sorry, last minute, sir. My video is not starting, so but I'm asking. Yeah, suggestion to Mr. Hirandas, sir.
Sir.
You have plotted the organic carbon graph very differently from I have seen earlier so far from anybody. Because with depth you plot it like upside down into the depth, and also depth need to be represented on the y-axis and the content on the x-axis. Then it makes sense.
Okay, sir. Thank
Your
you.
content is going up. That's giving a totally different kind of idea to the viewer.
Okay, sir. Thank you for the suggestion.
That's how it needs to be.
Actually, he is taken from somewhere, but I think that too from very means unrated journal. I think he has taken the material because without statistical analysis and some data is there, and something is as you pointed out, that depth as well as the content. So maybe, but he is learning. He will get more idea with your comments. Thank you.
Thank you, sir, for your suggestion.
Okay.
Okay, anyone else from this session? R. Rajkumar?
Yes, sir.
Yes. Shall I share your presentation?
Yes, sir. Thank you, sir. Respected chairman, co-chairman, and convener of the session, and other dignitaries. Good evening to all. So this is Raj Kumar, faculty in soil science and agriculture chemistry at Agriculture College and Research Institute to the Pondicherry UT, the Union Territory of Pondicherry. So I am here to present the topic sustaining productivity of Chinese potato cowpea-based cropping sequence through biochar in lateritic soils of Kerala. Actually, it was my PhD work. So laterite and lateritic soils covers nearly 65% of the total geographical area of Kerala. Means actually generally these soils are acidic, low in CEC, bare saturation percentage, poor in inherent fertility and high P fixation, and also means other problems like compact P horizon, reduced soil volume, low level of organic matter, or the other problems. So here this can be overcome through application of organic manures and amendment, and but the problem here is it is actually under tropical condition where a high rate of mineralization and soil compaction is the problem, which necessitates the continuous application of manure. So here here comes the role of biochar, which is an amendment highly resistant to decomposition. So instead of using other organic manures, we can go with biochar. So just the study was designed with an objective to to study the productivity of Chinese potato cowpea-based cropping sequence under the influence of biochar. So two crops were taken in sequential. So first one was Chinese potato variety Nidhi to study the direct effect of biochar and other treatments, and the residual crop, that is second crop, was cowpea was raised as second crop to study the residual effect. Actually, the treatments were 3 levels of biochar, FIM at 10 tons per hectare, then biochar with at 10 tons per hectare with and without soil test based POP. So soil test based POP package of practice was given along with biochar and without biochar, and these all treatments were applied only to the first crop and to study the direct effect, whereas in the second crop, in the same layout, without applying any fertilizer or without making any disturbance. The field was prepared and copy was taken in the second crop. So the soil belongs to Balapoya soil series and fine loamy kyolinitic isohypothermic typic plinthustals as per the USDA classification. So here are some photographs of field experiment showing the application of treatments. First, that is main crop and residual crop. So first crop with Chinese potato and second crop with cowpea. So to study plant height, average tuber girth, dry matter production, yield per plant, and tuber yield were accounted towards interpreting the direct effect of biochar and Chinese potato. So here the soil test based POP recorded the highest value for plant height and average tuber girth and dry matter production, which was comparable with soil test based POP plus biochar. So both the treatments, that is soil test based POP and soil test based POP plus biochar, recorded higher values with respect to these biometric observations. Whereas in terms of per plant yield and tuber yield per hectare, soil test based POP plus biochar registered higher values, that is around 147.98 gram per plant yield and 24.04 tons per hectare as against the control value. That is the lowest value in the treatment. And regarding the residual effect, that means for quantifying the residual effect of biochar and other treatments on a crop, we have taken cowpea as the test crop. Here the growth components like plant height, number of pods per plant, dry matter production, pod yield were recorded. And here again the treatment, that is soil test based POP plus biochar, which recorded highest yield in the that is first crop, again proved its proved best in the second crop also. So here also the values of plant height, pod length, number of pods per plant, diameter production, and pod yield were highest in this treatment, that is soil test based POP plus biochar. So actually this is just only results are given, and we have done this correlation and stepwise regression and path coefficient analysis. Significant and positive correlation were obtained between the growth component, yield, and soil properties, which confirms that the crop yield increase is a consequence of soil fertility improvement in terms of physical, chemical, and biological, especially pulp density, porosity, water holding capacity, chemical properties, especially pH, organic carbon, and available phosphorus content, potassium content, calcium content, and biological property, especially the dehydrogenase activity, were actually had more influence on this yield. And further confirmations also were arrived at from the stepwise regression, where we can see that growth components could be significantly predicted by soil properties in this experiment. And also path analysis we have done. Path analysis also showed that soil properties and tuber yield, path analysis between soil properties and tuber yield revealed that the properties like organic carbon, microbial biomass carbon, phosphorus, available potassium, calcium, and these properties had direct influence on the tuber yield. So as per the post-harvest analysis data, these properties had superior effect on biochar. They showed a superior effect of biochar and soil properties, so which can be identified, which was identified as a key factor in yield improvement through path coefficients. So coming to the conclusion, the study revealed the potential of biochar as an amendment in the highly weathered, nutrient-poor, acidic lateritic soils of tropics, especially Kerala, and the overall improvement in physical, chemical, and biological soil conditions through biochar sustained the plant growth and yield, which can be evidenced from the residual crop of this study. Thank you, sir.
Thank you, Dr. Rajakumar.
Sir.
What was the pH of the biochar you used?
Sir, pH was around 10.01, sir.
Okay. So what was the source of that biochar? Means
actually coconut husk and shell was taken in equal proportions, so that is 1:1 ratio.
And that was having such a high pH, 10 point.
Yes, sir. Actually, biochar means due to this pyrolysis process, attains high pH, means because of alkaline earth metals present in the charred material.
Okay, but with this pH, when you are adding to the soil, maybe your soil is acidic, then it is beneficial. But on the other hand, it must be having certain elements like calcium, magnesium, that too in high amount, that then only, or the sodium, that may have influenced the pH.
Sir, actually, sodium content is very less. Actually, calcium content, potassium, and magnesium, calcium, magnesium, potassium, phosphorus content were more in our biochar, sir. Even actually, there are means actually this study is in acidic soil with pH around 5.1, but there are so many literatures actually showing the effect of this alkaline biochar in even alkaline soil, sir. Even in alkaline soil, alkaline biochar through other ways actually it is promoting the yield. Means there will not be that much increase in the pH because the mechanism is different. So even the alkaline biochar can be up to pH 10, we can apply in alkaline soil. There are some studies. Means when I searched for this literature survey, I got like that, sir.
How this Chinese potato is different from Indian one?
Sir, actually Chinese potato means mainly I think it is because of size. I think so. Means it is actually the earlier name was different. That is coleus, tuber coleus. I don't know why the name was renamed to Chinese potato. Maybe the size actually it is means small in size.
The variety is the Indian one, or
yes, sir, it is variety is Indian one maybe that is released from CTCRI Trivandrum, sir, Central Tuber Research Institute, Trivandrum.
Okay, okay, thank you. Any query from participant? Dr. Hadda, sir? Dr. Bhardwaj?
Yeah. So, Dr. Rajkumar, can I ask you a question?
Yes, sir, sure, sir.
Yeah. So, the soils for carbon are improving the nutrient transformation, nutrient physical chemical properties.
Yes, sir.
Organic matter is an important component and important amendment, right?
Yes, sir.
Yes. So, why biochar? Why not farmyard manure?
Sir, actually, means that again the resistant means biochar, though it contains high amount of carbon, only a fraction of carbon is labile and remaining is highly resistant to decomposition, sir. Means so it's means it will actually another problem is we can't apply a material with high carbon content to soil. If it has labile carbon, we can't as such apply to the soil because it modifies the CN ratio and definitely it will have a means a negative impact on nutrient transformation. Because we can go only with the narrow CN ratio that is around as per the organic menus. Organic menu should come is around 20 to if it is less than 20:1 only we can apply to field. But here it one side it is having very less amount of labile carbon, so it will take care of the microbes, and another side it is having high recalcitrant carbon, so it will take care of the carbon sequestration in field.
It is also inert carbon. So it will not take care of the exchange properties of the organic matter, which we usually get from the other organic matter sources.
Yes, sir. But but it is having sufficient labile carbon, which that means as that of the FAM.
No, but it doesn't have enough labile carbon. The life of carbon in biochar is several years multiplied by the usual organic matter. It will stay there for hundreds of years.
Yes, sir. Actually, means biochar it has suppose if it is 60%, means around 65%. I means I will just take my biochar as an example. The biochar which I used contain around 65% of carbon, but the same actually the carbon the 65% of carbon was estimated using TOC analyzer and I got that, but the same biochar was estimated the carbon in biochar was estimated using chromic acid wet digestion method that is suggested by Barclay Black method, where I got only 5%. That means this 5% of carbon is labile carbon, which can be means met. Even
so
I actually there was
so the 5% was the carbon which was not pyrolyzed. 60% is the carbon which was pyrolyzed, and usually carbon in the organic matter is 60 less than 60 50 60%.
Yes, sir, less than 60%. But here the materials also is reduced in almost in composting everywhere. The nutrient content is not increasing. Actually, the material bulkiness is reduced. So that is why the composition is getting increased, sir.
Okay.
Dr. Rajakumar, sometimes we have seen that in many of the studies for checking the pollution in soil as well as water, they are using biochar as an amendment to, as they have the biochar is having high absorption capacity as well as they have the surface area they can absorb. So application of these biochars in soil sometimes may reduce the availability of nutrients which are available in the soil because the adsorbing sites are there due to biochar application. These adsorbing sites can be saturated with these the nutrients required by the plant, and when the biochar degradation is slow,
Yes, sir.
then the availability will be reduced. So
Yes.
can this hamper the means our our approach that application of these may improve carbon, but on the other hand can reduce availability of nutrients.
Yes, sir. But actually, it means the nutrients which is in higher concentration or otherwise in toxic, maybe means we usually the micronutrients are more affinated to the this organic manures. So I think the heavy metals or other nutrients may get absorbed, but these nutrients even
no, the biochar cannot. The biochar cannot see that what to absorb. It is the charge which matters, and the charge is negative, and most of the positive ions that which are labile in nature will get absorbed.
Yes, sir. Actually, means it will absorb means whatever means even that is actually the power of soil means that is CEC we used to tell it as a storage of nutrient that is negative charge. So though it actually absorb when means there should be equilibrium between the soil solution and the exchangeable sites. So when soil solution depletes, definitely from the exchangeable site it will come to the solution and it will meet the crop demand.
Yes. Very nicely explained. Thank
Yeah,
you.
another question I have is regarding your poster, Mr. Rajkumar, Dr. Rajkumar.
Yes, sir.
Is that you're presenting only yield
Yes,
of the crops, but in the conclusion, you're concluding that because of changes in chemical, physical,
yes,
and
sir.
biological
Actually,
properties they have, but there's no evidence in your poster about the chemical, physical, or biological changes.
sir, actually, I was expecting for oral presentation. If it was oral presentation, I could have included those things. Since it is a poster presentation, I restricted myself to this values, and I just wanted to tell that we have done miscorrelation path analysis. All confirmed that through soil properties, these changes are there.
But
So,
you could have taken out number of pods, pod length, DMP,
yes.
etc. Because nobody is interested.
Sir, actually, the title is productive. Since it is productivity, I focused on this. So while concluding, I added that point also because without means through that only these things came, and so to tell that biochar made this effects, I wanted means actually I added these points.
All right.
Thank you, Rajkumar. We will look for oral presentation next time, and we will have a good discussion on biochar.
Sure, sir.
Thank
Sure,
you.
sir.
Thank you.
Thank you, sir.
So it was Dr. Rajkumar. Anyone else? Prantika Kakati? Prantika? Balvinder Singh?
Yes, sir, I'm present here.
Sir, Prantika Kakuti is there.
Okay.
Sorry,
Okay.
sir.
Balvinder Singh also is there, sir.
Okay, Balvinder, you have your presentation with you, or you have sent?
Sir, my side is sharing.
Okay, share it.
Okay, sir.
Hello, sir.
Sir, poster on there. Screen, sir. Poster on there, sir. Hello. Hello, sir. Poster on there, sir.
Yes, it is visible.
Okay. Thank you, sir. Good evening, respected chairman, sir, and co-chairman, sir. Today I am present to hear my poster presentation topic is utilization of bale waste for animal feed productions. So is this I'm basically belong to ICRCIS as SRFs, and here I'm present to poster, and the small introduction has belong to a huge quantity of fruit and vegetable waste are generated from the various place of institution and bale crops. So according to Food and Agriculture Organizations, to 55 million tons are bale fruit are generated, and fruit and bale vegetable waste are also generated. And it is here recycling and also production the various type of animal feed like that enzymes waste This is my small objectives. The study report utilizations of bale based for the production of animal feed enzymes using a consortium of 7 microbes. So in the basically methodology, we have isolated the 7 microbes from the soil samples, and these microbes were degraded the pectinolytic, cellulolitic, and amylolytic microbes, and we are isolated then from the soil samples, and the bale waste was collected from the ICR CISS Lucknow, and it was the homogenized with the water and grinder, then added the 1 ratio to 1.5 water, and after that waste was autoclaved 121 degrees centigrade for 15 minutes, then included with 7 culture consortiums of bacteria cultures. These bacteria also submitted at NCBI with SSN numbers, and these microbes is basically Bacillus species, Bacillus pacificus, Bacillus marcoides, microbacterium, and Sphericus, Lysine Bacillus sphericus, etc. Acinetobacter citizendae also, and these bacteria are much more degraded, the pectinolytic, cellulolytic, and amylolytic. So here I'm after inoculated, the bacteria was inoculated in this mess form of bale waste. So and incubate at 35 degrees centigrade for the 10 days periods. Those samples were withdrawn at the 2, 4, 6, and 10 days intervals, then analyze the pectinolytic, pectinase, cellulase, and amylase activity as per the AOSC methods. And here we have observed the results. The 2 days maximum activity was observed for pectinase 454.0 units per ml, then cellulase 4.51, and amylase also 483 units per ml. After 4 days and 6 to 10 days, after that he was decreased day to day. And here we have prepared the animal feed supplement enzymes. These supplement enzymes is also used for animals as a digestive systems, and much more aparata like that diseases is controlled. And this is the crude enzymes also used as a small quantity, and this is crude enzymes basically. And these crude enzymes were prepared to bale waste, and this is much according to AOSC we have prepared. And this crude enzymes we have prepared to precipitate with acetones, then acetone removed by distillation units after centrifuge, and pellet form is coming, then dissolve in acetate buffer, and after that
conclude.
Yes, sir.
Give conclusion.
After the conclusion, we have observed the study related the bale waste could be successfully utilized for the use as substrate for the production of animal feed enzymes, and within a span of 48 hours, yield was maximum of 10. This is the following to references to AOSC 1995. So thank you very much, sir.
Thank you, Mr. Balwinder.
Okay.
Any query? Is it cheap to obtain the enzyme from bale waste, or there are other sources from where the enzymes can be?
Sir, bacteria also included in this one because bale waste is used as substrate, then bacteria in growth, and bacteria also produce the enzymes.
So the major production was through from the bacteria, na?
Yes, sir.
The bale waste was just a substrate.
Yes, sir. Yes, sir.
Okay, thank you.
Thank you.
Sir.
Any query?
No, sir. Myself, Prantika Kakoti. Actually, you called my name, but I was not
Okay.
there. Okay.
Yes,
sir,
yes.
Can I start? Means.
You share your screen.
Okay. Sir, I have sent my mail, poster in mail, sir. Sir, can you share?
You have just now you have sent it.
Not just now, sir. Before half an hour.
Uh,
Okay.
and that too in PDF format. It will be difficult to display. Okay, I will try. You stop sharing from your end.
Oh, okay, sir. Okay, sir. Sorry, sir.
Is it visible to you?
Sir, it's not my presentation, sir. But I have sent before half an hour, sir. It's sir from, it's like Anupama Das, sir. It's not mine.
Okay.
Sir, kindly check.
But it is from your mail.
Yes, sir. Yes, sir.
Please see in both the mails you have the same P25. No, okay, you have earlier also. Okay,
Yeah, P26.
okay, P26. Okay, now it is okay.
Yes. Thank you, sir. So very good evening to one and all present here. I, Prandika Agoti, is going to present my poster on the topic of influence of lime farmed manure, wood ash, and rice stubble management on the soil acidity and nutrient availability with or without rice stubble. So recycling of rice straw without causing any pollution to the environment, rather it improves the soil condition, is a challenge. So good management of the nitrogen and phosphorus showed the better yield in case of rabi crop, and it was reported earlier. And with the use of easily available and accessible mixture of lime plus FOM plus UDAS, it's attained a neutral pH in acid soil and in the rice fallow, and a good physical and chemical condition of the soil, and attain a balanced nutrient availability. This is the main purpose of our study. So in our study, the main plot is allocated with the rice stubble. In one case, the rice stubble is removed, and in another case, the rice stubble is incorporated. And in evaluation of the FOM, lime, and UDAS mixture is compared with the first one in unfertilized plot. Second one is where we have applied the half lime requirement plus FOM. And in third one, we have applied the half lime requirement plus FOM plus S. And in case of fourth one, the recommended fertilizer dose for tomato, that is 75, 60, and plus FOM. So we have analyzed many parameters like pH, NH4, NO3, phosphorus, potassium, aluminum, and exchangeable acidity. We have seen in this table the pH is increased due to incorporation of rice straw, whereas with increase in pH, the exchange acidity and aluminum become decreased, and it's due to a presence of LAP protein, means leucine aminopeptidase, that is coming from the decomposable rice straw in soil. That's why pH become increased. And in case of nutrient management, you can see where we have applied half lime requirement plus FOM plus UDASH showed the higher level of pH and lower level of exchange acidity and aluminum, and it's due to replacement of iron and aluminum from the exchange side of soil through the calcium that is coming from the CaO, means quick lime we have applied. And in case of ammonical nitrogen and nitrogen, both are significantly decreased due to rice straw incorporation in earlier stage, that is vegetative stage. It's due to immobilization of the nitrate nitrogen and ammonical nitrogen due to higher CN ratio, and later at 56 days of planting it's become increased due to lower CN ratio. And in case of the nutrient management, where we have applied half lime requirement plus plus FOM plus UDES, will show the better result in case of the ammonical nitrogen and nitrate nitrogen, because with increasing pH, nitrate nitrogen become increased because nitrifying bacteria become increased, and in due to release pattern of nitrogen from the organic manure like FOM will increase the nitrate and ammonical level. And in case of the phosphorus and potassium also, it increased due to the decomposition of the rice straw. It will add the phosphorus and potassium at the later stage. And in case of the nutrient management, the half lime requirement plus FOM plus S showed the higher level of phosphorus and potassium due to increase in pH through liming, and also the nutrient release capacity of the FOM and UDES. It's increased the increase the level of phosphorus and potassium in the soil. So lime and FOM mixture with the FOM, lime and UDES with FOM component provides the feasible nutrients to the soil, and it's compared with the, or it's better or cost effective than the recommended fertilizer dose. So thank you, sir.
Thank you, Prantika.
Thank you, sir.
Any query from participant? Okay, if not, I have one query.
Yes, sir.
Rice stubble management.
Yes, sir.
What was the size of the stubble? Means at what height it was being cut, by machine or by hand?
Sir, by hand, and the height of the straw was, sir, 35 to 40 centimeter.
Okay. How much time it take to degrade or decompose this?
Sir, it almost, yeah, sir, it almost takes, sir, 40 to 45 days.
But if we are going to amend the stubble with, say, some organic material, or to enhance decomposition, or
Yes, sir.
through some microbial cultures, then
Yes, sir.
can we enhance or reduce the time frame for decomposition?
Yes, sir, it can be reduced. Because, sir, if we apply the microbial composition, then what will happen? The urease and dehydrogenase and acid phosphatase acid phosphatase activity will be increased due to, sir, so organic source of the carbon from that is coming from the rice straw. And, sir, we have applied FOM, that is that pH of FOM is 7.87. So at this pH, the urolithic bacteria become increased. Means the activity of urolithic bacteria become increased. That's why it will take the lesser time if we have applied the microbial population.
Okay.
Sir.
In some of your studies at Jorhat, they have
Yes, sir.
used microbial culture for decomposition. Have you gone through that?
No, sir.
Okay. As a student, you should know some of the things when you are working on stubble management, that
Yes, sir.
the approach is microbial approach can also be one of the option for enhancing degradation and
Yes,
decomposition.
sir.
It will
Yes,
be
sir.
helpful in recycling the nutrients.
Yes, sir.
Okay, thank you.
Thank you, sir.
Yeah, can I ask a question also?
Yes, Dr. Bhardwaj.
Yes, sir.
Yes. So Prantika, I want to ask you this thing. So what is the visualized role of ash mixing with the FYM? So what role does this ash is play? What is the role this ash playing in this particular treatment? FYM ash, which is 2 kgs per hectare.
Yes, sir. Sir, actually, S provides, sir, mainly potassium and calcium, magnesium, trace amount of calcium, magnesium, and it also enhances the disease resistance capacity of the crop.
So, but it's only 2 kgs per hectare. 2 kgs per hectare.
Yes, sir. Actually, sir, we have to apply the S 3 ton per hectare. But sir, to reduce the cost of the farmer, sir, we have applied the S as a 2 kg, and we have applied, sir, FOM, sir, more, because FOM is more available than S.
But even FYM 2 tons per hectare is very difficult to apply. We go for 10, 15 tons per hectare, and when it comes to trials, then it is very small amounts. This amount is not very significant in terms of physical amendment or changes in the total amount of volume of soil you visualize with the addition of organic matter, for example, rice straw or wheat straw or everything else. So these are very small amounts, right?
Yes, sir.
Until unless they are very rich. So what is the percentage of nitrogen in your FYM?
Sir, it's like 0.36, sir.
See, and you adding 2 kgs, then there's a lot of changes.
Actually, I think this is basically just as a substrate for the organism already present in the soil, just to provide them the substrate. Otherwise, FYM 2 tons for a hectare area, it will be means a very in nutrient terms, it will be very meager. So
Yes. That is true.
Okay, but
Yes, sir.
on
That's
the other
fine.
hand, to reduce the cost, you can, but you have, for that you have to standardize. Otherwise, some of your studies might have standardized these that FIM 2 ton is sufficient to do this thing in the soil, in the acidic
Yes, sir.
soil.
Sir.
So that can be, but the main focus was the stubble recycling. So
Yes, sir.
maybe the what you told, that the to enhance the microbial activity for decomposition can be it can be used.
Yes, sir.
Okay, thank you.
Thank you, sir.
Thank you.
Yeah, thank you.
It was MSc.
Yes, sir. Yes, sir. I am a master student, sir.
Okay, good work.
Okay,
Yeah.
okay.
Thank you, sir.
Thank you. So next presenter, Mamta Prajapati.
Sir, I am audible, sir.
Mamta, are you available?
Good evening,
Yes,
sir.
yes.
Sir, I am audible.
Yes, you are audible.
Okay, sir. Sir, please share my poster presentation.
Okay. Just 2 minute time.
Yes, sir. Good evening to everyone. My poster presentation topic is studies on seed-borne microflora. Seed is the most important and critical input for crop production. About 90% of the world crop are propagated by seed, including some fodder crops. Pathogen-free healthy seeds is required to maintain desired plant population and good harvesting. Harmful microorganisms such as fungi, bacteria, virus, and nematodes can be carried with or in the seed, resulting in tremendous yield losses. The management of these pathogens during seed-borne phase is considered to be the key pest disease and management strategy. This is the objective of my study: isolation and identification of seed-borne microflora from different fodder crops. In vitro evaluation of antifungal activities of fungicides, nanoparticle, cupric oxide, and zinc oxide against seed-borne microflora of fodder crops. Fodder crops, they are grown for animal feed and food. Methodology: in this project, different fodder crops taken crop production unit like that Barcine, cowpea, Clautoria, cluster bean, maize, stylo, sorghum, and oats were collected from seed processing unit. These seeds are 2 years old, 13 and 14, 2013 and 14. Nanoparticles also used for treatment. So cupric oxide, they are making a 5 ppm, 10 ppm, 15 ppm, or 20 ppm dilution are made for this study. And zinc oxide nanoparticle dilution preparation solution, same 5 ppm, 10 ppm, 15 ppm, and 20 ppm. They are average particle size EPS 240 nanometer, molecular weight at 81.38, and fungicide here used Beveston. Name is ancient name is carbendazim. Then there are pictures spore germination or microflora, just like that fusarium and aspergillus. Fusarium, aspergillus, and curvularia seed spore germination show are less in 5 ppm and 10 ppm. They do not consider that all ppm as measure are considered to all type of microorganism retard the growth of spore germination. So they are concluded to here. We concluded that application result of the present study also revealed that different concentration cupric oxide and zinc oxide nanoparticle but maximum retard pathogen spore germination on all the treatments of fungi. Thank you, sir.
Thank you, Mamta.
Yes, sir.
Any query from participants? Okay. Nanoparticles, cupric oxide and zinc oxide, are these nanoparticles?
Yes. So they are in powder form and just for taking a minor quantity and stock, then prepare a working solution like 5 ppm, 10 ppm, 15 ppm, 20 ppm. They are also a nanoparticle.
In different concentration it is okay, but nano means something different. I think our nano expert is with us. Dr. Bhardwaj will
Actually, that is perfectly fine. Yeah, I'm here. So one of them is like 40 nanometers and other one is 240 nanometers. But I saw that, here in India, we using nano term up to 1,000.
Anywhere where there is nanometer.
Yes. 1,000 nanometers also.
So actually these are the chemical compounds, but average size zinc oxide say 240 nanometers, I think. Okay.
Standard definition, international definition says that nanotechnology should involve any particle, any dimension which is within 0 to 100 nanometers.
Okay.
Yeah, 100 nanometers is the limit.
Okay.
So but yeah. Can I ask this question? Yes.
Yes.
So I have we have nanoparticles which we use in this thing, but we have different forms of nanoparticles which we make. Miss Mamta, yeah, but this one, and we do not see much interaction between the microbes and the nanoparticles in our tests. There's no as such any effect of the nanoparticles on the microbial flora or microbial fauna, but you are getting. So that means that these nanoparticles would have some kind of toxicity or interactions with the humans also, if they are carried in the food chain, isn't it?
So this type of nanoparticle purpose only here used minimizing spore germination of seed adhering microorganism. They do not after harm to crops or fodder foods for animal feed.
But how do you know? How do you know that the animals which eat that fodder will not have any harmful effect of these nanoparticles? You don't know, right?
डॉक्टर भारद्वाज कह रहे हैं कि अगर नैनो पार्टिकल से ट्रीट करेंगे सीड को, फिर सीड जर्मिनेट करेंगे तो नैनो पार्टिकल्स उसमें जा सकते हैं उस पौधे में, और वो पौधा चाहे ह्यूमन खाए या एनिमल खाए फॉडर, तो उसमें भी चले जाएंगे, क्या वो हार्मफुल तो नहीं होंगे?
तो समटाइम्स हार्मफुल होंगे, तो ये इसका ड्रॉबैक है।
मतलब जैसे आपकी फंगल या स्पोर्स को वो खत्म कर रहे हैं या उसको जर्मिनेट नहीं करने दे रहे, वैसे वो जो एनिमल उसको खा रहे हैं उसको भी तो हार्म कर सकते हैं वो।
उसके गट ऑर्गेनिज्म को अगर
खत्म कर देंगे तो उसकी पूरी डाइजेस्टिव
डाइजेशन
एक्टिविटी
जी सर बिल्कुल अदर
मतलब ये
टाइप डिसऑर्डर्स
ये
आर
तो सिर्फ
इंक्लूडिंग।
हाँ ये बताया इसलिए जा रहा है कि आगे कभी आप प्रेजेंट करो तो ये सारी चीजों की जानकारी होनी चाहिए, है ना?
जी सर।
ठीक
Okay.
ठीक ओके, थैंक यू
थैंक यू सर इन्फॉर्मेशन।
अनुपमा दास, you are here, Anupma. Okay, anyone left with the poster presentation in this session can unmute and share your slide. I think no one has left. Anupma Das. So we are ending.
Hello.
Yes, Anupma, you are there.
Hello.
Yes.
Good evening, sir. Good evening, everyone.
Good evening.
Am I audible, sir?
Yes, very much.
I am Anupama Dash. Good evening, everyone. And today I'm going to present my poster. And the title of my poster is Soil Acidity and Nutrient Availability as Influenced by Organic Manure Sources with or without Rice Stubble. So as we know that rice is the main cultivated crop of our country, and most of the rice stubble after harvest of the crop is removed from the field or it is burnt. So if we can incorporate the stubble before the next crop, we can supply the nutrients which is inside the rice stubble. So to observe the effect of incorporation in rice stubble and the influence of 5 types of manure sources in incorporation or in effectivity of the rice stubble, we have carried out our carried out our study, which is a lab study having 3 replication and factorial CRD. There is a mistake, sir. This will be factorial CRD. And after completion of our incubation study of 90 days, we have observed that
this mistake is because you both, Prantika and Anupma Das, have similar study and similar treatment. That's why this mistake occurred.
Almost, sir. And I have carried out the field study also with split plot design, so that.
Okay.
Okay,
Continue. Please continue.
And after 90 days of the incubation study, we have observed that rice stubble incorporation has no significant effect in pH, but it has decreased the soil exchange acidity significantly. Among with the manure sources, poultry manure has increased the soil pH highest compared with the other practices. And as the our initial pH of the soil is acidic, so it has moderated the soil.
इसको जरा वो कर दीजिए.
Hello, sir.
You continue.
So it has moderated our soil pH. And it is observed that poultry manure and vermicompost has also increased the availability of soil NPK. And ammonium nitrogen and nitrate nitrogen has significantly increased the is reported to be significantly increased with application of a recommended dose of fertilizer, which has reported the highest effect. And but poultry manure and vermicompost has also shown effect as per with the recommended dose of fertilizer. Here, the rice stubble incorporation has slightly reductive effect of ammonium nitrogen and nitrate nitrogen, but it has also after 90 days, it has also increased the soil ammoniacal and nitrate nitrogen. This is because of enhanced soil enzyme activities. We have studied the soil acid phosphatase, urease, and dehydrogenase activities at both 28 days after planting and 98 days after the incorporation. And in both cases, recommended dose of fertilizer along with poultry manure and vermicompost has showed the higher enzyme activity than the other practices. These treatment effects of the treatments were explained by the previously study done by Mirza et al. On the activity of soil microbes, and the study done by Antorius et al. On soil enzymes. So after this study, we can conclude that poultry manure along with vermicompost can be a suitable option to foster the recycling of the rice stubble and betterment of crop yield and soil fertility in rice fallow soil. But further work should be done on the doses and enrichment of the compost. Thank you, sir.
Thank you, Anupma. Poultry manure was acidic in nature?
Sir, no, sir. The poultry manure, the initial property was slightly alkaline.
Okay. But here we say that for salt affected soil, we have promote that to use poultry manure because of acidic in nature.
For salt affected soil, sir?
Okay, it depends upon the feed of the
decomposition.
No, feed of the poultry.
Oh, okay, sir.
Any query from
Sir,
participant?
one query, sir. Excuse me,
Yes.
sir.
Yes, yes.
Yes, sir. Actually, sir, when we were going through our TOT trainer exam of Skill India, they have mentioned that we should not use this poultry manure in our vermicomposting. Is there any side effect of this poultry manure, sir? Is there any content of that create adverse condition for soil?
Anyone to reply? Long-term use of poultry manure is not healthy. It sometimes cause problem. So long-term use is avoided, is not recommended. But for once it can be used, that too the quantity should be standardized as per your soil, as per the availability of the material. And sometimes there are reports that it causes some problems in certain soil, especially under paddy condition where submergence is there. So there are certain changes which can be in terms of gaseous forms or something like that. So don't have much idea, but if Anupma or Prantika or Dr. Nile can explain much more better than, because they have been using these amendment for the acidic soil.
Thank you, sir.
Thank you. Anyone left for presentation in this? Anupma Das has completed her presentation. Anyone left in this session? I think 2-3 authors have requested that they will be able to deliver tomorrow. Then we have considered it due to their some engagements. So I think we are at the end of the session, if nobody is left with the presentation. So thank you all the participants. Thank you Dr. Bhardwaj, Dr. M.S. Hadda, who have been there all the time during the day. So we will be going for the second day tomorrow with the technical session 3 at 10 a.m. So I request all of you to join us for the technical session 3 with the same link, and all the participants should join, because on that basis only the evaluation committee will decide also not only the best poster and best oral presentation, but best participant will also be decided for both the days, all the 3 days. So you should be present during the sessions. Thank you very much.
Sure, sir. Thank you so much. Thank you so much.
So we are ending the meeting. Any comment from Dr. Hadda, sir? Okay, thank you. Thank you, sir.
Okay, sir. Thank you.